Our next presenter and topic will be Bastian Nießing from the Fraunhofer Institute. From the Fraunhofer Institute in Aachen. He will tell you about his institute and his department. And he will give us an insight into automating cell and gene therapies, automating the whole hot stuff on the market right now. Welcome, Bastian. And
hello.
After that, I would say we are clear where the market will go, and then we get real-world insights. So this is right now the ideal world, and Rainer afterwards will tell us how, or this is the ideal world, I must say, and Rainer will tell us how the real world will work. Yeah. Bastian, stage is yours.
Thank you. Thanks, Tristan. Thanks for having me. Thanks for the nice introduction. As Tristan already mentioned, I am Bastian Nießing. I'm from the Fraunhofer Institute of Production Technology, located in Aachen. I am the head of the department Bioadaptive Production, and I'm responsible for all the topics around cell and gene therapy in our institute. So today I want to give you a little introduction to the Fraunhofer world, to what is cell and gene therapy in general. Then I do a deeper dive on the therapeutics, then all about the automation, and then in the end I will have a deep dive into the collaboration we have with Haro Höfliger. So Fraunhofer Association, so I think most of you have heard about Fraunhofer, but I don't think that everybody knows what the concept is about and how large we are exactly. So we was founded after World War II, when the government said, well, we need a research association that is on applied research, so moving things from universities into the industry. This was what the goal for Fraunhofer is, and this is what we do until now. We take things from basic research and bring them into the market, into the industries. So we are the largest research organization within Europe. We have more than 8 independent affiliations worldwide. We have a huge network all over the world. In Germany we have 76 institutes with about 30,000 employees and an annual research budget of 3 billion euros. And this is what we do. All those different kind of institutes, they have different focus. So we are in production technology. There's also one in Stuttgart. This is also in production technology, but we are located in Aachen, and this is our institute. We was founded in the 1980s, and there is everything about production. So we came from automotive and aerospace, and for more than a decade we are now doing biomanufacturing. So we don't develop the process itself, but we care about the production of the processes. And we have over 500 employees, and from those 500 employees, nearly 10% are working in the field of cell and gene therapies, and annual research budget of about 30 million euros. And also there about 10% of this budget is within the field of cell and gene therapies. So what we do in my department, we do end-to-end automation solutions for our customers in a small scale. Whenever it comes to large-scale production side, we cooperate with Haro Höfliger. We do the whole digitalization of the whole process from end to end, and we have also invented a low-code software where you're able to have the completely process chain in your own hands, and you are able to put the things together and let the machine do what you want to without being too deep in the programmation of all the devices. So in generally, this is what Fraunhofer is, and now I want to go a little bit deeper in cell and gene therapy. So I don't know how many experts are here in the room today, but hopefully after this presentation you can call yourself an expert. So cell and gene therapy, how does it work? What is the important thing about this, and what are the points we want to trigger when it comes to therapy? So everything starts with the DNA. So your body is out of cells, and in each cell you have the same core with the same DNA, and only by the production of different proteins, the cells go in a different direction. So there are the differences between your skin and your liver cells only because of different type of proteins produced by the cells itself. So in each cell you have the same DNA, and this starts with one fertile egg and then it goes to your human being you are now. And this is important to understand, because once we are able to change the DNA or to force the cell to produce some specific proteins, we're able to deal with some kind of illnesses, and we're able to engineer the cells and what they are doing. And this is all about what we do in cell and gene therapy. We are forcing the cells to do what we think is the right way. So, and there are many different types of cell and gene therapy, but it's all on forcing the cells to do what we want to do. And with those proteins, we have different kind of cell types and different kind of proteins, but it's always the same. We start with the DNA, then we get the RNA, and from the RNA is then formed the protein, and we're also able to deliver the RNA to the cells. This was what we've done with the corona vaccines. And whenever there is a damage or something goes wrong with this process, it often causes some kind of illnesses. And so the aim of cell and gene therapy is to repair those defects within your DNA, or to enable or modify your cells, for example, to detect cancer cells. And so whenever this happens, there occurs an illness, and for example, cancer could be one of those illnesses. And when we are able to repair it on the very basic level, so the DNA, we are also able to cure the disease, not only to care about you, but also this is all what we're doing. It's all about curing diseases and not only taking care of this while having drugs. And therefore we have also two major fields when we talk about this. This is the cell therapy and the gene therapy. The cell therapy is all about editing the cells and optimizing the cells or changing the cells, and the gene therapy is all about working with the genes itself, so replacing or editing the DNA to force then the cell again to do what we want to do. So this was really basic overview of what is cell and gene therapy in general, and now I want to have a deeper look in the advanced therapeutics. So ATMPs, we've heard this before from Tobias, is all about advanced therapy medicinal products. This is the overclass of all kind of products which are in the field of cell and gene therapy. There are a lot of challenges whenever it comes to handle and whenever it comes to production. So they have a high cost because you have on the one hand side quite complex production process. On the other hand side, you're working with living materials. You have a lot of quite expensive compounds to keep the cells happy, and therefore it's quite cost extensive. You have highly manual processes, so all of the products which are at the moment in the market are produced in a manual way. There's no fully automated system with moving robots around. This is only in our heads, so there's no factory like this, like Tobias showed in his slides. We have a difficult regulatory framework, so we have in each country there are some changes. We have the huge markets on the US side, we have a huge market on the European side, but there are different frameworks and it's making things quite complicated, and all the processes are not that scalable because mostly they are for manual production, and when you have a manual production process you can't scale it really up, you only can scale it out, and outscaling is not that efficient than scaling up. So in general, ATMPs are divided in 4 different groups. We have the cell-based immunotherapy. This is maybe the most famous one, as there are the CAR T cells, for example. Then we have the cell therapies. This is all about providing different cells to the human body. So here we have the induced pluripotent stem cells, which are quite important. We have the gene therapy, where it's everything about changing or editing genes in your body. So There are different techniques for this. You can deliver it with the CRISPR-Cas9 system. You'd have like viruses which then deliver the genes, and you also can do it with the LNPs, so liquid nanoparticles. So there are different techniques evolving. And then you have the tissue engineered products, which then are conglomeration of many cells. Then we're talking about tissue, and there are, for example, cartilage tissue that could be printed to the size of a defect you have, for example, in your knee, and then it can be implanted. Also, artificial skin is the most famous part of this tissue engineered products. So now I want to have a deeper dive on two specific cell types, the induced pluripotent stem cells and the CAR-T cells. So induced pluripotent stem cells are stem cells or in general cells that were forced to go back in their evolution. So we call it reprogramming. So we start with somatic cells, so cells from your skin or cells from your blood. Then we add the reprogramming factors, which are showed here. So four of it. Mr. Yamanaka has discovered them in 2006. He got the Nobel Prize for this. And then we force the cells to go back in their development to stem cell. So stem cell is where it all started, and once we have reached this cell type, we're then able to do the differentiation in each direction we want. So we start with a blood sample, we go back to an induced pluripotent stem cell, and then can go, for example, forward to heart tissue, forward to brain tissue, forward to whatever kind of tissue we want. And then we can do on these cells, for example, experiments about blood screening, but we also can give them back to you to make any kind of treatment. And also, quite important is that we do on this stage the genomic editing. So there we can edit the DNA of those cells, and then because we started then from one cell again, we have then in all of the other cells we produce afterwards the same DNA. We have an annual growth rate of about 10% within this market of induced pluripotent stem cells. So now I came to the CAR-T cells. I think most of you have heard about this. CAR-T cells are cells which are against cancer, at the moment against blood cancer, but they're also researching in solid tumors. So we just compare those two type of therapies. On the one hand side, we have the chemotherapy, which is the state-of-the-art therapy. There's a cost of 20,000 euros per cycle. Normally you do multiple cycles, and then in the end you have a chance of healing or not. And if not, you are able to get the CAR-T cell therapy, because this is only allowed as a last stage therapy. And here we have the cost about 200 to 250,000 euros for one doses, so which shows that it's quite expensive. But the good thing is you only need that one doses. So it's a one-time shot, and then the cells, because they are your own cells and they live, they stay in your body, and your immune system will keep them alive. And therefore you only need this one shot. And there are patients that were treated like 10 years ago, and they are still free of cancer, because those CAR-T cells are able to detect the cancer. This is what we do outside the body. We enable the cells with the CAR receptor. CAR is for chimeric antigen receptor. So we put some DNA in the cells, force the cell to build up the CAR receptor, and with this CAR receptor, then the cells are able to detect your own cancer cells. This is the clue all about it. We take your own cells, change the DNA of them, give it back to you, and then they have new functions. And with this, I want to show you what is all able right at the moment, and what can be, or what is the good thing all about cell and gene therapy. And we can think about treatments of cancer, genetic disorders, autoimmune diseases, and even tissue and organ repair, unless we get the challenge to have a state production of all of those products. So market-driven, of course it is. We have large market in the US, we have large market in the Europe, and we have large market in the Asian area over here. We have different regulatory affairs for those, and so this makes it also difficult for us to have all those regulatories in the right way. So global market is also quite growing. We have annual growth rates of about 80%. And when we have a deeper look on the market size and how the market is shared, we see it on the pie diagram down there. We see that half of the whole market size is within the CAR-T cells. So, and the rest is divided more or less equally to all the other cell and gene therapies. And so, because the market is growing and there is a higher demand of those cell therapies, we need a scalable production solution. And this is when we think about automation, because we need to have a scalable production, and this is what we thought could automation solve this problem. So here I want to show you the typical production process, or not production but innovation process for new therapies. So we have on this side the production efficiency, and here we have the time, and down here we have the different phases when we develop a new drug. We normally start always within the R&D phases. So this is typically in universities or in an R&D department of pharma company. And then we do everything quite manually, which is completely fine because you have to do a lot of trial and error. You do like this and that, and you test a lot. And this is quite in a manual way. It's quite efficient because you have highly skilled human operators. Maybe they do their PhD thesis on this or something like this, and this is completely fine. Then you maybe have found something that could be a new drug, that could be heal something, or that could be used for something. And then you go in the preclinical phase, so you go in the animals, test it a lot, and there's also quite manual, and this is also quite fine because again there you have many back and forward, testing this, testing that, and changing the system sometimes, which is also quite fine. But then you go into the humans. And once you go in the humans, you have to make a decision on your process, because then you have to freeze down the process itself, and it's hard to change it afterwards, because the regulatory affairs say once you have a validated process, you're not allowed to change it anymore, and if you want to do it, you have to revalidate the whole process. So this is when we start the discussions with the regulatory affairs, if we're able to change from a round compartment to a rectangular compartment, because it's easier to handle. And sometimes they say no, and then we have to build an adapter to be from a round to a rectangular adapter, but don't change the shape of where the cells are growing. So just that simple things are not able to change. And this is where we as a research institute always say you have to think about automation or of production in a very, very early way. Otherwise, it's hard to have in the end a scalable and fully automated production process. So because what happens, you go into the humans, you have phase 1, phase 2, phase 3. Phase 1 you have about 10 patients roundabout. This is quite handleable when with a manual process. Then you go into phase 2 clinical studies, you have about 100 and 500 patients. So this is quite getting challenging if you're still working in a manual lab with like about 2 to 3 people. And then it comes to shift from phase 2 to phase 3. There happens 2 things. First thing is, when you're quite good, you get a lot of money, because some big pharma companies see, well, this is a good product, we will buy it. And you have to go in phase 3 for about 1,000 to 5,000 patients. So this is also where we think about, well, if I have here my team with 3 people and they have to produce like 3,000 doses, it will last like 10 years. So this is not what you want when you have a lot of money and you want to enter the market. And then they came sometimes to us, sometimes to other companies, and we taught to them, how can we do a system change? How can we have this manual process in an automated way? And then we end up with a partially automated production process, because we have to think of all the manual processes. In the end, we have only a compromised solution. And this couldn't be avoided if all the people would think about automation on an earlier time point within the product pipeline. So now I want you to show what we and our institute have done so far in the field of bio production and automation. So we did it for more than a decade. So we started all with the induced pluripotent stem cells. We have there this in the so-called stem cell factory, where we can produce in an automated way the induced pluripotent stem cells. And also we added recently the automated genome editing to this platform. So we're also able to automatically genome edit the iPS cells. We have one platform for the mesenchymal stem cells. So they're a little different to the induced pluripotent stem cells. We have one platform also for the automated production of CAR T cells. We have the CRISPR-Cas9 technology automated, and we have standardized protocols to do all the things in an automated way. So again, I want to show you those 2 cell types, iPSCs and CAR T cells. So on the iPSC side, we need an efficient and standardized production. And on the CAR T cell side, we also want to reduce the cost, because you can imagine, once those therapies enter the market and you get it for like first stage application approval, the health system will not be able to pay for those patients 200 to 200,000 euros per patient. So we have to reduce definitely the costs. So as I mentioned, what did we here so far? We have a fully automated genome editing process. So on the left-hand side, you see the CRISPR-Cas9 enzyme, which is able to do a very cheap and precise editing of the cells. We have in the middle the stem cell factory, and with this together, we have shown that we are able to have fully automated genome editing. So here is the gene PSN2, which is within the Alzheimer research, and we have here the indel weights. So we have here an automated and here the manual, and what you can see here is that we are at least as good as the manual process. But when you have a deeper look, you can't see it here on the slide. But if you have a deeper look on the standard deviation, we're even better than when you do it in the manual way. We've also done it with some other genes. Also worked quite good. We also did cultivation experiments, and here is also one thing what I want to show you. For those who haven't worked with cells, this is quite typical, because cells are living materials and they grow not equally, although you treat them equally. So it's because my cells will behave other than your cells, and this is also quite difficult when you come from an engineering point of view and you're used to work with a piece of steel, which is always exactly the same, and now you have to work with living cells and they are not exactly the same, makes things quite complicated. So for CAR T cell production, I've shown you here the whole production process. We start with the so-called apheresis, so we get a blood sample from the patients, then we do the selection and activation, then we do again the genetic modification, so we add the CAR to the cells, then we do the expansion. This can last 3 to 12 days, so here again you see you have long time within the machinery, you have long production times. Then we do the formulation and cryopreservation, and afterwards we freeze the cells down. Then has to be a lot of quality controls, release testing and so on. This lasts again like 2 to 3 weeks. and once everything is fine and the product can be released, we give it to the patient. So in total, you need about 4 weeks for this production process. Bad thing is that some of the patients die within those 4 weeks, and then you have to waste all of your product. Another bad thing about this is that, again, this is fully manually, and there are 5 companies who will or who have one of those products in the market, and the waste rate of those production processes are about 30%. So every third of your product will be just going to the trash, and this is also from a production side of view a nightmare, because you can imagine when you come from automotive and there is like the Mercedes-Benz down here, and each third car would be just go to trash, this won't happen. And you see how expensive they are. Okay, so what did we here? We have developed the 8-pass platform, which is all about AI integration and automation of the CAR T-cell process. So we have divided the platform into 2 different sections, the manufacturing section and the quality control section. And while automating all this into one platform, we're also able to do multiple patients in parallel. Multiple means 12. And on the manufacturing side, and then also doing the quality control, and therefore also have a higher yield of the platform, because of the different devices are used in more frequent way. And then we put artificial intelligence on top of it to have process optimization. So those are familiar with all the regulations will say, well, this won't be allowed. Yes, won't be, but this is not our was not our task in this project. It was more to show that it's in general possible. And so therefore, with automation, we can save cost of goods. We can have a higher quality and consistent quality, which is even more important. We can scale the processes, and we can trace the whole data over the whole production process. And therefore, we need the automation within the field of ATMP production. Now, I want to present you the collaboration. So why I am standing here, what are we doing with Harro Höfliger? It all started 2 years ago, where we, or even more, 2.5 years ago, where we met on a conference. And today we're in this fruitful collaboration. So what we can offer is, from a scientific side of view, the translation of your production processes into a machinable or readable production. And what Harro can offer is to build a large-scale machinery for any kind of process. And this is tried where 2 worlds very merged together, and this is very fruitful, because, as I also mentioned before, we do applied research, so it's our aim to go with things from research into industry, and now we're able to go really into the industry. So this is all about what we do in our collaboration. We want to combine our know-how and therefore enable all the pharma companies to produce their ATMPs in an automated way. And therefore, we developed the Happy Cell Factory, which was just launched yesterday. And here I want to show you some images of the Happy Cell Factory. So we have it, of course, it's all aseptic, so we have a huge isolator on top of it. We have here on the front the liquid handling module, where we can do all the liquid handling. This is all about CAR T-cell production. So where we can do also the harvest and the seeding of the cells. And then we have on this larger background part those smaller rooms. We call them hotel rooms. And this on the back side is the expansion mansion, because there will the cells grow. And this looks like this. So we have a lot of single hotel rooms, and on each hotel room there will be 1 patient. and you can just plug and play if there is some defect and change it. And then we have here an entry system, which is then feed the different patients with media, and also can take probes on a defined time point. And this is what you can see over here. And then we have there again the liquid handling station, and also the filling station, where we can do whatever kind of liquid transfer is needed. And this whole process is for bags, so let the cells grow in bags. But you also can imagine that we can change the different parts here within our hotel rooms to do like other cell cultivation. And with this Happy Cell Factory, I've done some calculations we have planned so far. So in the last 7 years, there was 5 different companies who produced those CAR T cells, and they have applied so far about 35,000 doses of this CAR T treatment. And the Happy Cell Factory is planned for 25 patients per day. So yesterday we was working around the shop floor, and they was all about how many pieces per minute are coming out of their automated platforms, and we are here 25 patients per day. So there's mostly one per hour, but not like 500 pieces per minute. So it's quite low scale from an automated point of view, but quite high scale from ATMP point of view. And if those 5 companies has this Happy Cell Factory, they would have lasted 9 months to produce this 25,000 products. Then I did another calculation, which is more important for the manager. So if we say we can produce like 25 patients per day, and we sell it for about 250,000 euros per doses, we're able to have a revenue of 2.2 billion euros each year. And this is quite important to also think about those numbers, and to go to the management and say, well, we need to buy this Happy Cell Factory to get all the patients we can address really get produced. Yeah, and this is the Happy Cell Factory, which we able to tailored offer cell types. We have fully customized needs. We are fully automated. We are completely closed. We have one central user interface. We have integrated quality control, and the most important thing is we are fully GMP compliant. So this can be then really used for the automated production of any kind of ATMPs. And with this, I would like also to thank my team. So Laura and Julius will also be here, and thank you for your attention.